So it's my distinct pleasure to introduce this year's keynote speaker, Professor Petra Schwille, Director of the Cellular and Molecular Biophysics Department at the Max Planck Institute for Biochemistry in Martinsried. I don't want to take too much of her talk time, so I will introduce her very briefly. Petra did her PhD at the Max Planck Institute for Biophysical Chemistry in Göttingen with Nobel laureate Manfred Eigen. And it was really during these early days in her scientific career that she developed a technique that I think we're all very familiar with, fluorescence correlation microscopy. At the time, she applied FtsZ to study protein diffusion and dynamics in solution. Then she moved to Cornell University, where she did a very productive postdoc with Watt Webb, also a hero of microscopy. And she further developed FtsZ to study fluorescent proteins, lipid bilayers, and cells. She then came back to Europe, to Göttingen in '99, where she was a group leader and later promoted to the rank of full professor in Dresden in only 3 years. In 2011, she was named director in Martinsried back in the Max Planck for Biochemistry. And throughout her independent career, she has continued to push the boundaries of fluorescent microscopy. And I have no doubt that today's talk will illustrate that. Her seminal work has been recognized by numerous awards. And to just name 3, she received the prestigious Gottfried Wilhelm Leibniz Prize in 2010. She became a member of the German Academy of Sciences in 2011. And more recently, she became a fellow of the Biophysical Society in 2017. Thank you, Petra, for joining us today. We look really forward to your talk.
Yeah, thank you very much for this nice introduction. It's really a pity that I'm not here in Cambridge today, but we have the Cambridge weather outside, so at least this gives me a little bit of a feeling that I'm in England. Yeah, so I hope it does not come as too much of a disappointment that I'm not going to talk about fluorescence technology as a technology. Of course, it is, as you can see in the first image, very strongly used in my lab, and also correlation spectroscopy, which is my dear friend since my PhD times, we are still doing. But what I would like to talk today about is our ambition to get to the core of biology, because this is something that has been riddling me since I was a kid. I always thought about what actually distinguishes a living system from a non-living system. Obviously, human beings being the far end of a living system, but already smaller living systems are somehow different from the stone or things that you otherwise find in nature. And how did I get into this? Let me have a little bit of a detour at the beginning to motivate why I'm doing this kind of stuff, being, in principle, a trained microscopist or spectroscopist, and this is actually what I can do best. Well, this is because the many techniques that we developed, which are single molecule sensitive, they have been applied to biological systems of increasing physiological relevance, if I may say so, during the time in Göttingen and then also in Dresden, where I was hosted in the Max Planck Institute for Molecular Cell Biology and surrounded by cell and developmental biologists. And I was very much believing in the quantitative power of our techniques, in the ability to really measure very precisely on a single molecule level. And we've seen that in the previous talk very beautifully, how you can actually track molecules in a cell. But everybody who has done that and previously done it in a reconstituted system knows the frustration that you experience about the quality of your data. If you have, developed your methods in an in vitro setting, in solution, or, even be it fixed cells, but the less complex, the better, can only, lose the nerves once you do it in cells and find that you're measuring pretty different things every day, because the cell is a complex environment. And because at some point I was super annoyed by this, and also by, reviewer comments that, well, we have studied this factor, but we have not studied this factor at the same time because it's also important. I decided that I'm not really interested in, what biologists call a physiological setting, because this is too complex for me. I'm a physicist. I'm trained to, reduce complexity, because I'm used to solving problems only if you reduce complexity. Now, the problem with biological systems is that they are basically by definition complex, because they've been evolving since about 3.5 billion of years, and this evolution has always been accumulation of more complexity. They have become resilient, they have become competitive, and that's only because they have become complex. So although I think it is possible to speculate about something like a simplest representation of a biological system, it is very hard to, define it. So just to motivate it from a biological point of view, I mean, what has made biology as a science was basically the definition of, or the formulation of cell theory in the 1900s. And, you call it the 1800 in the UK. It's in Germany only the 1900, so 1800 something, right? So that was basically formulated. And they, the big accomplishment was to show that life does not come about by, a godly, spark that goes into mud, right? If there's life coming up in a muddy system, then there was life already in the system. So there are three big pillars of this cell theory, and we all know them very well. The first is that our living organisms are composed of cells. Well, that's just a definition, right? This is how we call a living system. The cell is the basic unit of life. And then the elephant in the room problem or pillar is that cells arise from pre-existing cells, because that does not really solve the question how life can come about at all. And if you think about it, I mean, it must have been a simpler system than the ones that we find nowadays. And the question is, how simple? And is there any chance to get to this simplicity by, scientific reasoning? So there are two ways of trying to reduce complexity in living systems. There's the minimal genome approach, which is a biotechnological approach, which is trying to, slim down the tax that we are paying if we want to express a certain protein to the living system that expresses it. So that's Craig Venter's big domain and many others, right? So to find out how many genes do we actually need for the system to still stay viable. And it has been a true accomplishment to show that you can, for example, take a microorganism and reduce the number of genes from 1,000 to 500, which is amazing, right? It's only half of it, but 500 genes is still, for a physicist who's trying to control all the parameters in their system, 500 genes is still a no-go. So our approach towards understanding biology from first principles is pretty much, driven by, the hydrogen atom approach from physics, trying to find the very, very smallest possible system that you could call alive. And that we all know can only, because there's no true definition of what a living system is, we have to basically define a couple of features that we are after, such as ability to replicate, such as metabolism, such as ability to evolve. Now, our approach is basically, well, we are not chemists, right? So if we start to build, or if we try the bottom-up approach, if we try to build a biological system from scratch, there's no way that we can start with atoms, right? In principle, you should, because this is what it all is made of. But we are not able to create units, functional units from very, very simple atoms. So we have to start with functionalities that we already find in nature. So what we do, our approach is basically to take functional modules that are already known to be functional, and these modules are very often proteins. And that brings us to a problematic position, because we have basically three big challenges with this approach. The first is the very obvious conceptual challenge that if you want to reconstitute, or if you want to create a living system from scratch, it's absolutely stupid to start with proteins, which are very likely only very late in evolution. But they are the only starting points where we can already find function, and function appears to be a very important pillar of living systems. And my belief is actually, and I hope I can convince you with my talk, that it does make sense to start with functions that are already complex. Once you've understood what they're doing, you can try to minimize them from there on. The next one is the technical challenge, which is everybody knows who is working with proteins to reconstitute them to technical systems or to in vitro systems. Very often they don't do that, what they're supposed to do in the cell. They are much more complicated to work with if you want to purify them and reconstitute them in controlled settings. Well, that's a technical challenge that can be met in many cases, and in many more other cases it cannot be met. And then, of course, there's the academic challenge. If you're, if you're in the biologist's domain, of course, you always are facing even if you find something really cool about a protein in vitro, about a function in vitro, biologists wouldn't buy it and tell you, well, that's nice, but it does not have any physiological meaning because this function is, only observed in very artificial settings. But this, I would like to address this question at the end again, because I do think that the functions that we find in vitro are by no way unimportant, even if they are not physiologically significant in the way that they are playing a big role in the very cell this protein has been purified from. Okay, that was a long introduction. Let's just try to go in the middle of things that actually work. And as I said, if we want to start about living systems, we have to define features that we admire or that we consider the cornerstones of living systems. And of course, they are important complex functions such as replication, but they're also more simple functions such as pattern formation, which is not only reserved for living systems, but it is very important for living systems. So living systems are able to unmix, to use energy to beat entropy, basically. They're able to create structures and maintain these structures over time in spite of the molecules diffusing and exchanging. All living systems start with a morphogenesis and with gradient formations, and even in complex organisms, the way that the embryos develop goes over pattern formation. And it has taken, there was this seminal paper by Alan Turing about the relatively simple chemical requirements for morphogenesis to happen, which basically means that you need only two molecules that are interacting and consuming energy, and that with two different species, you can actually arrive at relatively nice morphological changes at pattern formation if you have diffusion and reaction. And it has taken quite some time for such a simple system where you have really only two molecules interacting with each other to produce biologically relevant patterns. And one of the systems we've been introduced by theoretical physicists back in Dresden by Carsten Kruse and his colleagues. And Martin Lohse, a student at that time, he basically accomplished the purification and reconstitution of these proteins, which are actually the min proteins from E. coli, which are doing in the cell pole-to-pole oscillation, which is basically setting or positioning the cell division machinery by washing away the cell division machinery from the poles. They basically confine it to the cell center, and by this, guarantee a symmetric division. And these oscillations, which are basically here sped up quite some time, it's about a minute time scale. If you take these proteins out and reconstitute them on a membrane that mimics the bacterial inner cell membrane, then they form these beautiful waves, which are just a representation of an oscillation if you don't have any boundaries, right? So then, of course, there's no turning around. It will just spread out as waves. And if you look at these oscillations or at these waves in more detail, and here come our expertise in the single molecule world. If you look at the single molecules producing these patterns, you can actually see that they're in spite of you looking like a traveling molecules, they're not traveling. What travels is just the wave. So it basically is a assembly of a Laola wave in the soccer stadium, where the whole crowd stands up and sits down, and doing that successively gives the representation of something moving. So what happens is that the molecule that you see here, or that is labeled here, is a ATPase, which is basically a soluble molecule which presumably dimerizes or multimerizes upon ATP binding and goes to the membrane. So only once it is ATP bound, it basically adheres to the membrane, but then it cannot hydrolyze the ATP, but needs a helper protein, which is recruited only if a large enough number, so it's basically a cooperative process, this recruitment, they basically, a large enough number of membrane-bound MinD ATP will recruit its helper, its catalyst, and only when this guy comes, it will basically allow the ATP to be hydrolyzed, and then the whole machinery is released from the membrane. And because this is very tightly regulated in terms of cooperativity, you have this this traveling wave. You if you look at it, it's a hot zone. It's a lot of membrane-bound MinD, and once there's an Min-E coming, it will basically dissolve the thing almost like a catastrophic event. It looks very beautiful. We have looked at it with AFM, but I'm not going into detail here. Anyway, so this is the machinery. We know reasonably well how it functions, although the details are still not, some mysteries still remain. In order to get that into a reassembly of the in vivo system, of course, you have to log it in a compartment. And Katja Zieske, a student at that time, she accomplished that not by logging it in a real vesicle, that would have been our dream at that time, but by basically providing little bathtubs that look like bacterial cells and just leave them open and air on the top, and just the reduction of the volume basically fulfills all the requirements that is necessary for these waves to actually result in real oscillations that very much resemble the oscillations in the bacterial cell. It even grows with the length of the compartment. This is what it does in bacteria, because bacteria grow in only one direction. So the period of the oscillation during the growth of the bacteria changes, and only if the bacteria is large enough to divide, it's basically time to assemble the division machinery. And that's actually intriguingly, that also happens if you grow. Of course, we don't grow it, we just offer different sizes. Our compartments, you can actually see how the period of the oscillation changes, and at a certain length to width ratio, which is pretty much the same as in bacterial cells, you can see that the oscillations split up into daughter oscillations, and that's basically the time when the real cell would divide. Now, as I said, the presumable function of this oscillation is that it positions the division machinery, and the division machinery is a huge machinery of many proteins. At the very first place, however, it's FtsZ. It's a tubulin homologue, basically a filament forming protein, which is supposedly also able to not only create a ring, but also to somehow contract this ring. I will tell you later about the ability of this thing to actually contract. What we show here is that these min oscillations are indeed able to spatially regulate the positioning of these FtsZ filaments. Here they are in 2D. The FtsZ is only, they're not filaments, they're only little, very small filaments, all not any rings, but you can see how the min waves basically wash them away, and whatever the min waves has passed, the FtsZ binds back to the membrane. Of course, in such a setting, you would not get a static gradient, but if you log it up, you can actually, by the pole-to-pole oscillation, you can get a static gradient with a clear dip of the min proteins in the middle. And this is exactly where you see the assembly of this FtsZ protofilaments, not yet rings, because as you might remember, these are not closed compartments, they're only half shells. So this works, and I would like to remind you that this is really simple, the system here. We have only in there the membrane that basically mimics the bacterial inner surface, the two min proteins, ATP, and in this case then the FtsZ. Now, of course, since we are talking about proteins and you know, proteins as building blocks, as functional building blocks, of course, if we want to understand how these functions could be modified, we now can do mutations. And with the MIN proteins, especially my student Simon Kretschmer, he undertook, you know, a lot of different assays to mutate them, and found that in order to, you know, to fiddle with the pattern forming ability and change the patterns that you could get, and also the phenomenology of, for example, the oscillation, you would only have to introduce very few changes in the bacterial proteins. So the MIN-E, which is the catalyst, the inhibitor in the wild type form, this was just the patterns that you get in the oscillation phenotype. If you just take one amino acid out of replace one amino acid in the membrane targeting sequence of this, you can actually see that the wavelength of the patterns changes, and even more importantly, the phenotype of the oscillation would change completely. There's no pole-to-pole oscillation anymore. And if you terminate the ability to bind the membrane entirely by taking away the full membrane targeting sequence, then you can see that you get a very funny phenotype of oscillation, like a top-to-bottom oscillation, which would not make any sense in the real bacteria. And of course, such a mutation would not be viable if you would do it in a real cell. So what we also found, and here comes now I would say the really interesting, you know, conceptual part about it, because now we can try to reduce the complexity of our proteins, and really try to go towards minimal functionality. For example, in this MIN system, there's something that has been riddling us as physicists from the very, you know, first moment when we dealt with it, because in order to recruit, or in order to catalyze the ATP hydrolysis in MIN-D, in membrane-bound MIN-D, oh, sorry about that, the MIN-D has to basically switch the MIN-E, the inhibitor, into an active state. It's a conformational change in a latent state. It does not catalyze ATP hydrolysis, only in the reactive state it does do it. And the big question is why this complexity? Why do we need you know, why does the inhibitor has to be switched on? Why can we not just have a reactive inhibitor all the time? Because the theoretical predictions told us, we are working very closely with theoreticists, Erwin Frey and his group from the Ludwig Maximilians University here in Munich. They basically said, well, it should work also if it's always, you know, in an active form. So we tried to make a mutant which is always active, and we found that indeed in this mutant we still see pattern formation. However, the ability to form patterns is now much more critically dependent on the ratio between the two molecules. So if you have MIN-D and MIN-E in different mixtures, the concentration ratio that you can go for the wild type is relatively wide. However, for the mutant, where you have an always active inhibitor, the concentration ratio is much narrower. And that would, of course, mean for a biological system that it is much less viable and much less resilient if there was any factor that changes the concentration. So here you can see in principle for your eyes how evolution works, right? So in order to get the principal phenomenon of pattern formation, we don't need it. But to have a stable pattern formation, something that is also robust against changes, we need to introduce this additional degree of complexity of a conformational switch. But it's a good news for us as, you know, principle lovers to know, well, ultimately it's not necessary. Only if you cannot control the concentration, it would be necessary. And now you can even go on, because the MIN-E does not only have this conformational switch, but it also has two other features which we did not fully understand. One is that it is a dimerizing. It has a dimerizing, domain. And it also has a membrane targeting sequence. Well, I talked about the membrane targeting before. You saw that if we get rid of the membrane targeting, we still get pattern formation, although the phenotype of the oscillation looked weird. So we know we can do that one away, we can terminate the conformational switch. The ATPase stimulation helix, we cannot get away because that's the one that is talking to the other molecule, right? So that would be the ultimate, the smallest possible unit. If there was, in the perfect world, we would only have this one. But we still have the dimerizing domain in the real protein. So if we can do without this one and without this one, can we also get away this one? And it turns out, no, we cannot. We need at least one of the motifs, either the membrane binding or the dimerization, to complement the little ATPase stimulating helix. But the good news here is that one of them is actually sufficient. And the even better news is that this, the way the helix is brought to the membrane does not matter. And also the way the helix is dimerized does not matter. So we do not have to take the native dimerizing domain of min-E in order to get the patterns, but we can take just any dimerizing domain to do that. And that's really now very good news for an engineering approach. We can minimize the system in the way that we just put a DNA strand, a short DNA stretch of a couple of bases, and the complementary DNA to the other helix, and by this induce pattern formation in a completely hybrid and completely, in this case, really very, very simplistic min-E variant, right? So I hope I can show by this that, well, it does make sense to start with the complex proteins, although they are the end point of evolution, because if we understood them, we can try to re-engineer complexity and find what is actually needed and what is not needed, and even go back to much simpler systems. This was actually, I have to say, this was done by Tamara, and she's also a student in the lab, and this is at the moment submitted. Yeah, so many theoretical people who look at our data have always asked, well, that's nice, but the real Turing pattern formation is actually static gradients. It's not traveling waves, it's static gradients, and not only static if you have locked up the system, but also static if you have an infinite system. And a good news is, for the lovers of Turing patterns, that there's only a little bit that you have to change in the system to convert our traveling waves to standing waves, or even fixed static patterns. So if you would like to use the min proteins, if you could, to introduce patterns in downstream proteins, there's no problem to create these, polarity inducing systems. Now, of course, the question is, what can you do with min proteins? They are basically only, in principle, E. coli proteins, and they are trained to, interact with other E. coli proteins, and you can probably not use them to interact with any other protein. And the serendipitous finding that Bea Ram, an excellent student who's just moving on to her next career level, she found out that this is not true, that min proteins can in fact regulate completely different proteins from other organisms, and regardless of a functional relationship. How did she find that out? Well, she found that if you have sorry about that, let's go back. Well, these min proteins are binding the membrane, and obviously they are competing with other proteins binding the membrane. So if you have a peripheral membrane binding protein, for example, in this case, a super uninteresting M-cherry protein with a membrane targeting sequence, you can bring these M-cherry protein into the reciprocal pattern. So wherever you have min proteins, you do not have the M-cherry. And that is nice, not too surprising if you have a competition reaction. Of course, if you have more of one kind, if one molecule is a better binder than the other one, obviously you can, outcompete the at a specific position. So this is very nice, but not so surprising. The surprising part came when Bea asked the question, what happens actually if these proteins that are on the membrane cannot leave the membrane? cannot be, you know, just chastened away by the MINs. And then something really funny happens if you have a protein which is diffusing in the membrane, like in this case, a simple streptavidin with 2 membrane anchors, which can, however, not dissociate. Then you find that the traveling MIN waves are able to push these proteins out of their way in a almost pumping-like fashion. This is all, of course, an active process regulated by ATP hydrolysis. So it's an active pushing away, creating void zones, and by this, you know, establishing real gradients of membrane-attached molecules. The only condition in order to fulfill this is that these molecules have to be able to diffuse. If you do it with, you know, big fat transmembrane proteins, you cannot move them away. But anything that diffuses in the membrane can be brought into a gradient formation by the MIN proteins added to it. And if you ask why is this actually happening, it turns out that this is basically a fully new, in a way, process of pushing away molecules on a surface. So it's an active directional transport, which is not motor driven, not in the way that you have specific cargo by a specific motor, but it's basically still a mechanical process. How do we find that out? Well, we basically provide 2 different molecules on the membrane. In this case, it's boring for a biologist, but very nice for a physicist because you can control the size very nicely. It's DNA origami, huge bars of DNA attached to the membrane to, either by only 2 cholesterol anchors, so with a relatively small footage on the membrane, or with 15 cholesterol anchors with a large footage on the membrane. And if you subject both of these to MIN waves, you can actually see that this guy will be much more efficiently transported by the MIN waves than this one, simply because you have much more, you know, resistance and friction between them. So it's a real mechanical driving, almost chromatographic process, which we can explain. This is now something that we have developed with our theorists colleagues from Arvind Frey's group. It's called diffuseophoresis. It's a phoretic process, actively driven, basically on diffusing molecules. That has been described in theory in thermodynamics, but it has never been observed or even described for biological systems. But it appears that this is a very powerful way of transporting molecules directionally in a way prior to the development of molecular motors, which you do not have in this sense in bacterial cells. Yeah, so I've shown you that already. Okay, so I guess that was kind of demonstrating how nice it can be to do this in vitro reconstitution. And of course, you are taken away and you find all kinds of things that you have never dreamt of. But of course, we also have a goal, and our big goal, a technical goal in the lab, is to try to find a minimal system to actually accomplish a division of a compartment, because a division is really totally essential to living systems. Now we are working with membrane compartments, with vesicles, and it has been described very often that in order to divide a vesicle, it's actually not so complicated. So it does not really need to you know, involve all the complex biology and biochemistry that you have in cells. In principle, if you just have a little bit of, for example, a phase boundary between liquid ordered and liquid disordered state of a membrane, a little bit different viscosity, you can easily drive the system into division just because of the line tension between 2 domains. That has been described long ago, but this is a thermodynamic process. It's running down an you know, a gradient, a potential gradient. That's not super interesting, but it might help assembling a machinery that is actually able to divide the vesicle based on energy dissipation, which is what we hope to accomplish in the, you know, near future or far future, depends. Now, what kind of a machinery would you like to have? Well, obviously, if you look at division in cells, if there's, you know, usually an active regulated system, you usually have filamentous cytoskeleton elements that do the job. even if it's not yet understood for any of the cellular systems, how exactly it works. I mean, there are two big candidates, one from the bacterial world, which is FtsZ, which is the filament that I've been talking about, which is regulated by the min waves. And in the eukaryotic world, the most prominent system is the actomyosin system. People like to think of division rings that contract. We nowadays know that this picture is probably far too simple for the FtsZ, but also for the actomyosin. But for sure, what you need to have is the ability to position, the contractile machinery to a specific point or the specific zone, ideally the equatorial region. So that depends on many factors, whether or not you can confine the molecules. You have to attach it somehow to membrane, otherwise you cannot exert any force on the membrane. Many of these proteins are only active if they are bundled or crowded. And the big question in many of these systems is then, how could this system actually contract? Well, what I showed you here is actomyosin, which is pretty clear to contract. There it is more the problem of mastering the protein reconstitution in the vesicles, and we are now at the position where we can actually confine, very clearly defined ring systems to the inner surface of a vesicle membrane. What is missing here still is the way to, make these vesicles easy to divide. So I haven't brought data with me, but we can contract these actomyosin, but it always, what you see is that the rings basically, they glide along and then they contract somewhere, but of course they don't do anything to the vesicle. And that's pretty easy to, imagine. If you have a round ball, if you want to contract that, it's a relatively senseless effort, because if you want to, contract something, you better elongate it first. Something about FtsZ. So the interesting, FtsZ was very long our favorite molecule in order to try to reconstitute into a ring-like structure, because you can see that it forms rings in bacteria. And Martin Lohse in my lab, who basically did the min wave, when he was a postdoc in Harvard, he basically found that this FtsZ, if you reconstitute it to a flat membrane, usually it's not supposed to be on a flat membrane, it's supposed to be in a tube, right? Because the bacterial cell is a tube. But if you reconstitute it on a flat membrane, it basically treadmills. So it basically hydrolyzes GTP, and by this it basically goes around in circle, like you know it from microtubules, but not directionally. It is directional, but not point in one direction, but rather in circles. And that was very unclear why the molecule would do that, or whether the system would do that. What is the point of going around in circles? And the first question was, of course, why is it always the same direction? And Diego Ramirez, one of my students, he basically tried to find why this, this chirality was actually there. And he found that the chirality was due to basically 3 axes of symmetry of the FtsZ filaments. One axis of symmetry is obviously polarity. It's a polar filament, like a tubulin is also forming polar filaments. It also is curved, so you have one axis of symmetry from the bending, and then the third axis of symmetry is the membrane attachment. And if you turn around the membrane attachment and basically go from a left-handed to a right-handed symmetry by just changing the membrane attachment to the other side of the molecule, from the D terminal to the S terminal, you can see that the treadmilling goes around in the other direction. And what we also found is that this treadmilling, if you put these molecules on soft membranes, here we have supported membranes, so obviously there's no membrane deformation going on. But if you put these filaments, the FtsZ filaments, on soft membranes, like on vesicles, you can see that these guys who are basically treadmilling clockwise, they are basically doing indentations to the inside of the vesicles, whereas these guys who are basically anti-clockwise, they are sucking out the membrane, basically creating little little spikes. And that can only be explained if we, and that actually goes quite well together with some structural findings, if we assume that the FtsZ is not a flat, but rather a helical filament. So it does deform membranes, and it also forms rings. The problem is it does usually not form rings of the size of the vesicles that we're using. So what we have to do, and that's also what I basically told you about the actomyosin rings, in order to be able to position a ring such that it can actually divide something, what we have to accomplish is that we elongate our round balls into more, cell-like structures. And we are not yet there, but we've made a great breakthrough by showing that by microfluidics, you can actually do that. You can suck in a microfluidic channel, you can actually suck in the vesicles and deform them. And if we do so, we can see something very funny. This is also something we did not even look at the beginning, and then found that it's really interesting if we have a FtsZ decorated membrane and suck it in, and the FtsZ forms relatively large regular bundles, and suck it in the in this channel, and release it again, then these vesicles are, the bundles of FtsZ are completely changing their morphology. So they all form little rings. In the ideal world, we would see rings that are the size of the vesicle, but here we have to engineer a bit. But at least, it's getting there. Yeah, so regarding the FtsZ transformation of membrane. So the big question is this treadmilling that we observe from FtsZ, is it actually doing something to deform the membrane? Because there is this old dream that the FtsZ ring would be able to contract. And it never that has been actually really visualized, how an FtsZ ring can contract. Of course, it would be very beautiful to see that. And people in the in vivo scene, they basically even claim that the treadmilling is not doing anything in order to change the diameter. What it does is that it recruits cell wall building enzymes, which then do the job of reducing the diameter of the cell. Well, that's nice. It's probably a very reasonable, assumption. But of course, we don't want to get involved with cell wall building enzymes in vitro. So our question was obviously, is there not anything from this in treadmilling that indeed induces forces on the membrane? And Diego went on to assemble a really beautiful assay in which he accomplished the formation of very soft membrane tubes that are about the size of a bacterial cell, so much, much larger than the membrane tubes you usually have from optical tweezer experiments, about 500 nanometers in diameter, even closer to 1 micron. And if he does that, he takes the vesicles, large vesicles, you can see the little FtsZ rings, and he takes them and pulls tubes, very soft tubes out from these little treadmilling rings. And there you can then see the treadmilling is actually going on in this soft membranes. You have, you don't have this chirality anymore. You have clockwise and anti-clockwise treadmilling. And now look what you get once you pull the tube. You can actually see that the treadmilling leads to a deformation of the tube that looks like a spiral. And this can be basically imagined that by the chiral, the bidirectional treadmilling, you basically take the membrane and twist it like this, and that will actually result in a force that will be there to destabilize the membrane. Of course, this force is not sufficient to split a bacterial cell, all right? But it is there. And the nice thing about it is that the protein is actually able to produce the force. And of course, there are now many, many helper proteins out there to actually accomplish much larger forces, but the principle is in the protein, and that's important for us. What we also found, and that's another really serendipitous finding about the Min proteins, is after many, many years of trying, we finally got the Min proteins into vesicles. This is actually Thomas Litschel who accomplished that using a method C-dyes, where you basically centrifuge droplets in an oil-water system through the interface. And if you lock in the Min proteins in vesicles, these are real vesicles now, you can see that they do oscillate. And of course, here also we are not yet there to deform them into bacterial shape, although that's now in the make. But we see very clearly the oscillations. Obviously, we have not a clear pole-to-pole symmetry. We have many different modes. And very often we have actually the phenotype that there is an oscillation here. You can see it very nicely in this vesicle here. The phenotype that you have an oscillation between everything going to the lumen and everything going to the membrane. So, and these vesicles, if you now take them and under tense them so that they are very floppy, you can see that they start jumping up and down. Once the molecule, all the proteins binds to the membrane, it's actually tensioning the vesicle. And once it goes into the lumen, it's loosening the surface tension. By this, you basically have this very funny phenotype of bouncing up and down. And even more, if you now make them even floppier, you can see that they even do something like a division process, right? And there's no filament in there. This is just from the membrane, membrane physics that goes on, because it's if you change the local curvature of the membrane dramatically, you can actually see that they undergo, the transition to a, divided membrane. Now that's really cool. So you don't even need a filament. Of course, this is not very well controlled, right? So there you can never be sure that it really divides in the middle. But at least, I mean, you see that the principal phenomenon needs much less than originally thought. And if we go even one step further and try to bring it all together, min proteins, the oscillations that basically bulge out the membranes, and the FtsZ as a treadmilling ring, we get really crazy stuff. And we are not yet there to actually explain everything, but here, I mean, that I think can be best explained by the Jeff Koons balloons. So what we have here are basically loose membrane, vesicle-like structures that, as the min proteins come and go, will basically breathe and will be tensioned or shrunk. And if we then also have the FtsZ, we basically get something like, the balloons which are twisted and make all kinds of weird structures. What is also, and that's a bit more controlled here, this is my Fang Fu, a postdoc in the lab. She basically accomplished to produce membrane tubes that are hollow by making peptide crystals that are coated with membrane, and dissolving the crystals give you something like a membrane tube is basically spanned between different how would you call that, aggregates of protein lipid. Sorry about that. And if you now add the min waves, you can actually very nicely appreciate how these tubes are being pulled apart and relaxed. And here you have also a very clear mechanical signal that you can analyze and now really visualize how the force is produced from this min waves, which is, I think, very nice. It's a real mechanochemistry, if you will, which is not molecular motor based. Okay, with this, I'm at the end. Let me just, summarize a little bit what I would like to say about our work with reconstituted proteins, because I really do think that there's a lot of value doing this, not only to learn more about what the proteins are doing in the cell, but what they could do in the cell and what they or their predecessors in earlier stages of evolution were actually able to accomplish. So we have a lot of hidden functions out there, and I guess every reconstitution person knows that. Now, we also know that if we want to publish our fantastic data, that our biological colleagues tell us that, well, nice, but who cares? They might not be physiologically relevant, but I would like to take this around and say, well, they are relevant. And if they are not physiologically relevant, we have to find out when under which biological conditions they were relevant. So they might be very, very important in this, conceptualizing a minimal cellular system where you do not have 1,000 genes, but only a couple of them, and they might still be able to do the whole thing if you just release their functions. So these kinds of functions that we found in vitro, they are the ideal modules, or might be the ideal modules for a true bottom-up reconstitution of a minimal system. And with this, I would like to thank my people. This is a picture which is already 2 years old. This was a kind of a celebration, a birthday celebration, where I invited all my ex-people. Many of them are now group leaders or even professors themselves. And the people who have done the work that I've talked about were mentioned in my talk. And I should not forget to thank my collaborators, in particular Erwin Frey and his fantastic team of theoretical physicists, but also Dirk Trauner, who's done a lot with optical switching, our system. I didn't talk about that a lot. Germán Rivas from Madrid, he's very intensely collaborating on the bacterial system. And Carsten Kruse, I would like to thank, because he was the guy who introduced the MINS to us. And yeah, thank you for your attention.
Thank you so much, Petra. I wish we could all applaud and you would hear the applaud from the room. But in the meantime, we can start by asking a few questions. So there's not a lot of questions in the Q&A yet, but maybe I can start with the questions. I was really fascinated by these FtsZ spiral filaments that form in the GUVs and how they seem to move on their own. It's not, there's no ATP hydrolysis there.
It's no, it's GTP hydrolysis. Sorry, I didn't say that. It's a GTP, this is an active process. So basically you need GTP hydrolysis for this treadmilling to happen. That's like in the microtubules, basically.
And that's why they spiral in one direction, essentially.
Exactly. So they polymerize, and I mean, it's basically these filaments. They are basically growing on one side and shrinking on the other. And this is because you need to add basically GTP-bound, monomers that will, once the GTP is hydrolyzed, will dissociate again.
So how far are we from starting to make self-separating?
I actually think not so far. I mean, it's also a lot of other groups who are, very intrigued by this. So I think with the power of the many groups who are doing that at the moment, I'm pretty sure in about 5 years from now, we will be able to come up with a simple system that is able to, maybe even earlier than that, that is able to self-divide, actively self-divide, not just falling apart, right? So, I mean, falling apart, that has been shown long ago, right? So in order to destabilize a vesicle so that it falls apart, that's easy. But what we want is a active regulated system that needs energy dissipation to do that.
Yeah. But I think Maria has a question.
Yeah, it's going to be a very naive question. So this is fantastic. It's fascinating results. Thank you so much for the lecture. So when you have a cell, you have to feed this cell, right? Somehow. So these systems also need feeding with GTP, with the...
Yes, absolutely. Absolutely.
Okay.
So the metabolism part, that's probably the hardest of them all. I mean, in order to activate a living system, you need to have a very, very smart way of coupling energy dissipation to, entropic, reduction of entropy in the system. And of course, what nature developed there over the billions of years is so sophisticated. There's no way that we can come up with a good solution. I mean, we for sure cannot. I mean, there's a lot of good chemists out there who I think will potentially be able to create something like a metabolic system. I mean, even he has a quite a nice system to it's basically macroscopic chemistry. Macromolecular chemistry that you have to, invoke for that. But that is something we will probably not be able to solve ourselves.
I think, Jan, you had a question? No? Jan, no?
There is a question here in the Q&A, if I may read. So,
yeah, go ahead.
Rowena Cray says, in oscillations, did you notice specific frequency pattern specific to protein?
Yeah. I mean, the good thing about these patterns, I mean, the system is relatively well understood. So in principle, it depends all on the concentrations, on the K on, K off, on the, diffusion coefficients of the molecules. And this can all be modeled. So you can very precisely nowadays say, how will the patterns look like if you tune this and that in the system?
Could I maybe ask a question, if that's okay? Yeah, hi Petra.
Hi.
Really great talk.
Thank you.
Could you sort of maybe go even further? I mean, obviously, FtsZ, although that's simple for cell biologists, it's complicated for membrane biology. So,
yeah,
what do you think happened before FtsZ. Can you speculate on that a little bit? How you think that sort of, so the origin of life and the origin of cellular
yeah, so what is really interesting, I mean, if you stay with the membranes, I mean, of course, you can also speculate about, well, it probably doesn't even need a membrane. There's this fantastic paper by Jülicher and Hauer and Hyman about, that it could all have been a condensate and so, but let's stay with the membrane. So what is really nice, there has been one very interesting paper this year by Lipovsky and Dimova, showing that if you have a very, so like our bouncing vesicles, so you have a vesicle which is basically very, very floppy. And if you just bind GFP, totally inspecific, just GFP to the surface, the change in curvature by binding the protein will be able to bring the system over the energy barrier into division. So in principle, if you just accomplish a local change of curvature in a system, and if you accomplish to refine or localize this very defined change of curvature, you can accomplish that. So meaning that if you just have something that curves a membrane, however it looks like, either, of course, you always think about bar proteins or so, or dynamine, yeah. But if you just have something that induces a local change of curvature in the membrane and you accomplish a gradient formation for that thing, then in principle you can get there. Mm-hmm. I mean, that's of course something we would like to try, and we are also doing that. I mean, we are trying to do that with DNA origami, because DNA origami is very nice. You can design the curvature, and you can design the affinity very nicely, even better than with proteins. So our hope is that we can use the min oscillations to basically create a zone in a vesicle of, where they have a confinement of a curved protein. And if that already lowers the energy barriers to make the decision possible, that would be fantastic, right? It's too good to be true, so it will probably not work, but at least it could work.
I have one more question, if there are no other questions. I was also really intrigued by this displacement of proteins on the membranes, active displacement. And
yes,
I was wondering if one can speak of a force being generated.
Yeah, absolutely. Absolutely.
Any sense of how strong this force is?
Well, we haven't really calculated in terms of a force. I mean, but you could. I mean, in principle, it's just it's a gradient which basically results in a force. So it's a chemical force. You could calculate it. I haven't. We probably should do that, but we haven't, discussed it in terms of a force. But what is very clear is that it's a mechanical cue. Yeah. Which was not at all expected. So everybody talking out. Also ourselves, we always talk about the min waves as, chemical waves and not about mechanical waves, right? Yeah. Very, very strange.
So I mean, I can imagine lots of applications coming out of this. Is there a way of making this specific so you can choose what you push or not push?
Well, potentially, yes. I mean, you just have to attach something to the min proteins. I mean, but then it's not anymore mechanical. Then you would add some chemical things. I mean, what I think is really cool is that you can sort molecules according to their size, right? You can really do some sort of a two-dimensional chromatography, which is kind of cool.
Yeah, even in the cell.
Mm.
All right, I don't see any more questions in the audience, so maybe we can thank Petra one more time for a great talk and great answers. And